Today I've got a question for you. What do sea urchins, climate change, and a sushi ingredient have in common? The answer is they're all part of a story that could help either save or destroy our oceans. Today, once again, we're doing something slightly different because we're at Deakin University's Research Center, where they're taking us behind the scenes into the research that they're doing for kelp conservation, one of the key ingredients in climate conservation initiatives. So come on, let's jump in.
One of the misconceptions that we often find when talking to community is that kelp is a plant. And you'll probably hear me say plant today, so sorry for the confusion, but it's just the go-to word that we often use. But it isn't like a terrestrial plant that we can see out the window here. So the couple of differences is that the main one is it doesn't have a root system like our terrestrial plants have. So it has these hold-fast structures at the bottom, and that hold-fast, as the name suggests, it holds fast onto a rock substrate, and it's an extension of the tissue from the seaweed itself. And these little root systems here for this particular species is called haptera. But like a terrestrial plant, it photosynthesizes to grow. So that means it captures light and converts that light into energy, and it helps sustain the seaweed and grow from there. In doing so, through that photosynthetic respiration period, it then also gives out oxygen. And that gives life to everything underwater and helps sustain a macroalgae forest or a kelp forest where it might be situated. So it's hugely important for life underwater, but also for us on humans and why we should care about it. And well, it tastes really nice for those that might have had the opportunity to taste wakame, which is a Japanese kelp. That's one that's often in our diet that we might be able to have access to. But also it's super important in terms of our oxygen supply. So what I'm going to ask you to do is a little exercise. If you all take a deep breath in. And I'm going to ask you to do that again. So another deep breath in. That second breath is because of seaweed. So without seaweed, we are going to struggle to take that oxygen in. So even though we get a lot of our oxygen supply from our terrestrial plants, our marine plants are also doing that same role for us. So a little bit with the seaweeds and the trouble they're facing underwater, it's a little bit of out of sight, out of mind. It's under the water, we can't see that there's issues there, and we don't necessarily take action. And so today we're going to hopefully connect you a little bit more with kelp if you haven't already don't know about kelp or don't care about it, or you've seen it washed up on the beach and it's slimy, it's stinking, you've got to step on it to get into the water. What a hassle. Hopefully change that perception slightly as to how unique it is and how important it is and the threats that it's facing, but also leaving you with today with some hope on what we've been doing here on site in terms of restoring it. So with that, is my clicker going to work? Yep, the video. I'm going to introduce a unique reef system to you. You may or may not have heard of this. Has anyone heard of the Great Southern Reef? Yay, I love the nods and the hands up. Okay, so for those that may not have, that's okay because you're about to hear about it. This is a reef system that stretches over 8,000 kilometres of temperate Australian coastline. So it extends from New South Wales coast all the way across to Western Australia and including Tasmania. So 5 Australian states, bigger than the Great Barrier Reef, but who's comparing? And in that reef system, a lot of people are like, wow, that's all kelp forest. No. Important thing is that it's interconnected through all these different ecosystems. So it's not just kelp forest. There's seagrass beds, there's sponge gardens. There's a whole heap of different marine ecosystems that are interconnected through biological, physical and ecological processes and chemical processes too. But the one connecting figure of this system is the kelp. So if there is a macroalgal forest or a kelp forest, kelp are the common feature across this system. And one in particular, which is the one that we've been working on here at Queenscliff, is the golden kelp or Aclonia radiata. And you see some of the footage of that coming up here. And just while we're on the Great Southern Reef, the reason why, it's huge, it's quite big, but it's also really iconic and important for us Australians. We rely on it economically. If you look at the abalone, the crayfish industry, the tourism industry that connects those states, we need that system to be healthy for economic reasons. And if that's not enough to sell us, then in terms of us wanting to walk on the beach on a Sunday, go for a snorkel, scuba diver, surf, and do so in the condition that it's currently in, we need it from a social or cultural perspective as well. And then, of course, from an environmental point of view, to sustain what is considered to be a global biodiversity hotspot. The endemic species rate in this system is really, really high. And in fact, if you're looking at the seaweed endemic species, the red seaweeds alone, there's 70% of those red seaweeds are not found anywhere else in the world. So very, very iconic and special. But unfortunately, and you've probably seen it in the media, particularly over in South Australia for the past couple of weeks, the algal bloom that's had devastating impact, that this system is under threat. And some of those threats are varied, and it's very localised specific issues. And the one that we're going to talk to today is issues that are specific to Port Phillip Bay, but also recognising that there are lots of other threats that are facing this system at the moment. Oh, before I move on though, this is a decorator crab and the best dressed organism in the ocean. It actually can put things on top of its exoskeleton to camouflage itself. So it's quite a cute little thing. So back to seaweed. So a healthy kelp forest is something that might look like this, a golden kelp. Lots of golden kelp in there, but there could also be other species of seaweeds. It's not just one. And there's lots of organisms that live there as well. And specifically for Port Phillip Bay, what we're seeing is this healthy kelp forest still exists, but we're seeing this decline at a rapid rate. In some areas, there's a 90% drop of kelp forests in Port Phillip Bay. And the reason why that is because of this little critter down here, spiky creature, the purple sea urchin. Now, we'll preface that by saying that the purple sea urchin is native. It's not invasive. It needs to be here. We want it to be in a kelp forest, but we need the density to be at a balance that it's not tipping over the edge and eating all the kelp, because that's what they do. So this particular purple sea urchin, because of the bay's had these changes over time, and that could be from water quality, urban infrastructure that's put pressure on that system, climate change, you name it, different pressures have happened, that sea urchin has thrived in those conditions. And a sea urchin has the ability to reproduce amazingly. Thousands and thousands of larvae can be expelled from this species very, very quickly and very rapidly. So they can reproduce quickly, and in doing so, they are hungry, they're eating the kelp starts to decline. So we go from what we consider to be a good balance of density of urchin to kelp, to suddenly that urchin balance being doubled and the kelp are starting to disappear because they're feeding on it. Scientifically, that density has been looked at to be about 2 urchins per square metre is where we kind of want it to be. So anything tipping beyond that 2, you start to see the pressure of that system change slightly. And if those urchins keep persisting and keep feeding, unfortunately, what we then see it become is, and I'm clicking, is an urchin barren. And you may or may not have seen an urchin barren example in Port Phillip Bay. If you have gone snorkeling to some of these sites, what this would look like would be bare rock, lots of urchins in that place, rarely or next to nothing, no kelp in those areas. So these urchin barrens are an issue, and we're starting to see some of those urchin barrens persist in areas that have been there for decades and they're not returning back to a kelp forest state. And when I say returning back to a kelp forest, that can happen. So if it does become an urchin barren, naturally it can switch back to a kelp forest without us humans intervening. And that can be from lots of different reasons, but changes to that particular location from a physical, biological point of view. Perhaps a predator's come in and reduced those barrens, or the urchins themselves have died off for whatever reason. Then you can start to see that shift in that balance over time, the kelp returns and off it goes again. We've seen that happen in California. In fact, there was a site I visited 2 years ago where one corner of the headland had lots of kelp. Literally on the other side was an urchin barren. One year they had the fires, and then they had some strange heat water coming through, but then all the urchins disappeared. They moved down to deeper water, the kelp returned, and that site didn't need any intervention. They were just about to start doing restoration into that site, and they didn't need to. So we're still trying to understand those shift differences. So today I'm going to go through some projects that we've been working on at Queenscliff, and the first is the biobank one, which is like a seed bank for terrestrial plants. So you know, with all the fires and the droughts that we've been having, we often collect the native seeds of different species. So it's the very same concept that we've collected seed from kelp species along the Victorian coastline, and we're keeping them here for long-term storage in our facility. Another work is looking at the genetic or gene flow of the Aclonia populations. So we're basically trying to understand, say, if the population here at Queenscliff, how related to that is from populations of kelp down at Portland. And the reason why we want to know that is if we're restoring kelp, do we need to be restoring the same species from that location or the same stock from that location? Or can we develop a more thermal or resilient type kelp strain here in the lab to put out into those areas? So we're trying to sort of understand the mechanics of that, and it's something that hasn't quite been figured out yet in the restoration space. And then, of course, touching on some of the pilot trials that we've done with our partners from Parks Victoria, Nature Conservancy and University of Melbourne. We've done some restoration trials in 2 sites in Port Phillip Bay. But before all of this, I do need to add in a bit of science. So we're going to start off with the adult. This is the one that you might see when you're out snorkeling or washed up on the beach, perhaps. It's the one we're most familiar with when we think about kelp. So that's where the life cycle starts. Then on the tissue or the frond of the plant, you then start to see the reproductive structure appear. And this can appear any time of the year that we found in Port Phillip Bay. This can be variable depending on the location in Australia or wherever Aclonia is found. But here in Port Phillip Bay, we've been able to collect it any time of the year. We do find that after a rain event, it's not as great. It's already released, so it must be some sort of freshwater influx into the water that stresses it out and it releases the reproductive tissue. So this is a darker patch that will appear on the kelp itself. And that darker patch is, the scientific term for that is sorus or sori tissue. And you can often identify it quite easily just by touch because it's quite slimy. It's a slimier part compared to the other kelp. And it's darker, and if you hold up to the light, you can actually see a really darker patch. So that darker patch inside its tissue has, oops, I've gone too far, millions and millions and millions of these tiny microscopic zoospores. And again, we actually released these yesterday, and they didn't do so well. I think it's that time of the year. We've had a really hot summer and the reproductive tissue wasn't as great as it normally would be, but that's science. So we've got a little small sample to show you, but this is what they look like if you don't happen to see them under the microscope. They're tiny little circles, and these are what they start off their microscopic stage as a little zoospore. Here they haven't decided whether they want to be a male or a female yet. It's not until they get to the next stage, which is the gametophyte stage. This is where they'll either be a female or a male. And here at this stage, before they can fertilize and head on to the next one, is the female gametophyte will develop an egg on the end of the tissue here. The male gametophyte will release sperm, which is then attracted to that egg. And you can see that in action here. And it fertilizes the tip of the female gametophyte. And that female gametophyte then transitions into this early stage baby, adult or early sporophyte. So the adult stage is actually referred to as sporophyte. So there's 3 ones to remember: the zoospores, the gametophytes, and the sporophytes. So it's these little baby sporophytes, which are about 6 weeks old, is what we grow here to that stage at Queenscliff. We would then outplant them for any restoration from that point on. Also, in a kelp farming perspective, it's around about that time that they would grow them in a hatchery and then plant them out onto farm. So the biobank is super cool idea. This was off the similar project down in University of Tasmania, where they're biobanking the giant kelp, Macrocystis. And we thought we would have a crack at that doing with the Aclonia. So we were funded by the Victorian government to do that. And so what we have done is isolated the gametophyte life stage, so the boys and the girls, and we can keep them in this red light condition where they don't transition to the next life stage. And so they stay at these gametophyte stage and they vegetatively grow, but they don't sexually reproduce. And so that's really cool that we've been able to do that with this red light. We can keep them for as long as we like, and we like to say we've got kelp on tap because of that. But more importantly, we now have kelp from all these different locations along Victorian coastline, that if Port Fairy was to undergo some sort of issue, whether it was a hot summer, kelp all dies off, we've got the original Port Fairy stock here if we needed to restore that back in that site. But the sites that we've collected from around Port Phillip Bay, and these were our seed bank sites as well, if they're circled in a group, it means that they are genetically closely related to each other. So the Point Nepean, Queenscliff and Pope's Eye sites are all relatively similar genetically. Port Arlington, St Leonards are too, but then we've got these separate isolated sites up at Jawbone, Ricketts, and 2 sites over here in Mornington. So these differences are very small from a genetic point of view, but they are enough to be able to show that distance does matter. And so the St Leonards site, for example, is quite different to Jawbone. So then from a restoration point of view, should we be putting different stock from these different locations into these areas? And can we cross St Leonards with Coral Beach, for example, and does that make them more resilient by having these different populations cross together and grown up? And then our restoration projects, I'll finish up on what we've been doing here. So we've had 2 sites that we've looked at, which is up at Jawbone Marine Sanctuary and Ricketts Marine Sanctuary. And this was the multi-agency project that we've been working with. And it involved lots of different stages, but in a nutshell, what we needed to do here was to go in and remove all the urchins to the density that we wanted. So we're not removing all of them, just down to that 2 urchins per square metre. Once we'd done that, University of Melbourne were leading the monitoring side of it, so they'd go in and have a look at before and after the urchin removal, and then when we put our kelp out there, they would then go and monitor before and after of that as well. The Nature Conservancy, they got the good job. They got to deploy all the kelp that we grew. So they were the divers that went out there and very, very gently put the kelp into these sites and then monitor them for us as well. So Deakin's role really was to grow them here in the hatchery. And then after 1 year, the really exciting thing was not only did they get to probably about 35 centimetres, so size of a ruler, a little bit bigger, they also started to show signs of being reproductive, which is super exciting for us because it shows that we can step back. If they're going to reproduce and contribute to that population, we've taken the urchins out to the density that they needed to be out, and they can also reproduce and start to do their thing, and we don't need to keep putting kelp back into that place.
Thank you so much for watching. I hope you learned as much as I did and were just as fascinated, because that was freaking awesome. If you'd like to learn more about Deakin University and their work, make sure to check out the links below where you can follow them. And if you haven't done so yet, make sure to hit subscribe on this channel, where I will continue uploading cool content with everything related to the ocean, making it accessible to you. See you next time.